It seems like we have a really good mix of scientific people and policy people, so this is fantastic. I want today to be as discussion-based as possible, so if at any time you want to jump in and say something, that's great, and we'll be breaking into groups to do some exercises later on as well. So my name's Carla Davidson. I'm a postdoctoral fellow at the University of Calgary. Right now I'm working actually in vaccine research, but my background is pretty varied. I've worked in everything from forestry to wildlife to genetic engineering to experimental evolution to vaccine development and design, plant biology, quite a lot of things. And a lot of these areas have kind of been on the forefront of things that are very controversial or have a lot of relevance to policy making. So this is giving me a lot of interesting experience on how science is interpreted both at the policy level and at the public level. So this issue is what really first got me interested in how science is perceived at the policy level. And if you can't see, just tell me to move. I'm a walker. This paper was published in 1998 by Dr. Andrew Wakefield. And it was a small study looking at 12 kids and their associated gastrointestinal issues. And kind of hinted at an association with the MMR vaccine, which is for measles, mumps, and rubella. Nothing really earth-shaking, though it was a very small study. However, in a press conference following the release of this paper, he suggested that if parents were concerned about their kids getting autism, then they should consider getting a single shot of the measles vaccine instead of the lumped MMR vaccine. What he didn't disclose at this press conference was that, in fact, he owned shares in a single-dose measles vaccine that had no commercial value as long as public health agencies were buying the MMR combined vaccine. What he also didn't disclose is that he had actually documented some of these kids' medical records to make the association between getting the vaccine look like it was more of an issue than it was. So in the following 10 years, there were quite a few studies looking at the supposed link between MMR and autism, and nothing was found. And then investigations started into how Dr. Wakefield had done his research. And in fact, in 2007, the British government General Medical Council convened a council to look at his conduct and found that not only had he doctored some of these records, but he had subjected these kids to some very invasive and painful medical procedures they didn't need. So this included colonoscopies and lumbar punctures or spinal tap. And in fact, one child had complications arising from this study that gave him lifelong health problems, and this was a civil case that was settled out of court. In 2010, Dr. Wakefield's medical license was revoked, and the paper was retracted from the Lancet. So now, if you go to the Lancet website, this is exactly what you find. The paper's still there, but with a big retracted banner on it. So what's interesting and really scary about this issue is that despite all of the scientific evidence and despite the evidence of how badly done the original study was, parents have still been reluctant to vaccinate their children. And throughout the Western world, vaccination rates have been dropping over the last few years. They're starting to come back up. But these vaccination rates are also low. They tend to be low in clusters. And that's dangerous because once the rates get too low, you start to lose what's called herd immunity. And so now these diseases have a possibility to start popping back up. And in fact, that's what's happening. So what's interesting to me is where the skepticism is coming from. And the interesting thing is that amongst parents who refuse to vaccinate their children, they tend to be very highly educated and very skeptical. And these are good things. I'm a parent. I'm skeptical about anything that has to do with my child. But what I want to know is, why is the scientific evidence less credible than anything that you can find on the internet? And this is what started me asking these questions. So the point of today is that I want to be able to give you guys tools to be able to go to the primary literature, to find the scientific studies, and give you a specific set of tools so that you can read through them and start making some critical decisions on what you find valuable in those studies. Sound good? All right. So I started off by Googling, because I Google everything. And first I Googled scientists, and I hit images, and that was depressing / interesting, but there's a lot of pictures of Einstein. There's always like some Einstein-looking cartoons. There's quite a few pictures of mad scientists. That's a good one. There's a lot of pictures of people looking at things that are completely unintelligible, and they look very serious. And there's more people looking at things that are unintelligible, but now they have lab coats and rubber gloves, so that's good. But, you know, everyone here has been working with a lot of primary science. We know that the picture is quite a bit more diverse. There's obviously a lot of the stuff that we deal with are scientists who are out working in the field. And science is, of course, a lot more diverse. It's not just nerdy-looking guys anymore. And in fact, it's also an international endeavor. Science is a systematic way of examining the world that is practiced the world over. And now we're finding that these international partnerships, especially in the last 10 years, are becoming a lot more prevalent, a lot more well-recognized, and very, very important. So before we get started, I want to put the question out to you guys. If you could define science, how would you do that? Anyone? I will point at someone. Yeah.
So you gave us the answer.
All right, yeah. Let's turn to page 2. Yeah, exactly. So there's an element of observation, observing what's going on in the natural world. Might as well bring it up.
But decision support. Decision support. Science as decision support. So when I think of science as proving something so that I can make a better decision.
Okay, interesting. So if you were to go through that process, right, how would you start?
I'd probably find somebody that I trusted to give me a robust answer that would be unbiased.
Okay. Interesting. Okay, so there's issues that I'm picking up on that of evidence, right? Like essentially what science is there to do is to provide evidence for making decisions, right? So there's an element of observation, of experimentation, right? If you are looking for evidence, then a good way is to frame a question that you can test and go out and gather evidence. And fundamentally, it has to be repeatable. And once you start following the literature for a while, you find that this is actually a huge issue. It doesn't matter how high you get, that quite often a study will be published that no one else can repeat. And in fact, this is what led to a lot of the investigations into the MMR study, was none of those claims could be substantiated. But science at its base, if it's well done, should be able to be repeated by anybody who has sufficient resources. And it's systematic. Right? So if I'm explaining science to somebody who doesn't, isn't really in the field, I say, basically all we do is look at variation in whatever phenomenon we're in and try to attribute that to something. It's a systematic way of looking at variation in some natural process and categorizing it. Sound good? Okay, this one I didn't give you as many answers, but obviously in doing, when we're involved in policy decisions, there's a lot of elements involved. There's values, there's economics, there's the science as well. So I'm interested in hearing from you guys, how do you use science when you're making your policy decisions?
From what I've seen in my experience, is you rely on one person.
Interesting.
And that's sometimes scary.
Do you mean like one person as a conduit or one person's research?
Both.
Okay. So do you deal a lot with people who are doing primary research and say, go out and find out for me this? Okay.
Which is scary.
We'll talk about that a little bit later, but it's a big problem. Yeah, I agree. Yeah. Anybody else? So are you interested in looking at the science that tells you that something is going to be a problem, or are you more interested in developing, say, remediation type guidelines after the fact?
With the watershed science that we do, for example, the Bow Basin Council, I'm on the board of the Bow Basin Council, so we do watershed management planning. We look at science and in regards to river health to help us determine what are the limits and thresholds for fish, for example.
Okay.
So we do look at what's the best available science, but we do rely on our relationships with people that are working in the field for their knowledge of what is the best research to rely on.
Yeah. Oh, okay. So there's a lot of background data, right, that you're going to be using to make these guidelines, like those actual numbers, right? Okay. That's one thing. There's also an element of trying to do projections, right? Okay. So I see some nodding. Is that do that sort of thing with the city?
Well, my stuff, so in my old job, we do science. We look to see how things are performing, how we can rank things based on the studies that have been done, everyone has a new idea, but has that new idea been tested? Have the results that are being claimed, can it be proven?
Okay.
And then on the forecasting, looking ahead, okay, this is where we are now, this is what history's been to get us here. What would happen if we do this or this or this? Does it happen in other places? What was the results? And also contradicting claims that, this will never work because it happened to my cousin. Okay, well, was your cousin a one-off, or was it, or does the data support what happened to your cousin?
I like that one. I have a friend of a cousin of his brother, and yeah.
And those have a lot of weight.
Yeah, that's true. Okay. And so when we're talking about making projections too, right, there's two aspects of the scientific data that you have to rely on. It's not just the baseline data of where we are now, but our understanding of the processes that are going to project us down the road. Right? Yeah. Yeah.
I guess one of the problems that we encounter is that you almost can't go down the road of science because there's so many studies on this side that say, as an example, there's no worries with the use of pesticides. You can go over to this side and you can find a whole host of studies that say, yes, we are determining that there's stronger evidence of a link with pesticide use and certain diseases. And then you're automatically called junk science because it doesn't correspond with this other side.
Yeah.
So you're kind of in this key to okay, well, so
It's anything to do with toxicology and human health, you have my complete sympathy. Those studies are so complex. They're extremely complex. And teasing apart any sort of environmental variables that lead to whatever disorder is a huge problem. And for example, BPA, bisphenol A, that this is a stabilizer that's been added to plastics, right? And so research has started finding that, in fact, it leads to premature feminization, right? And it's basically an estrogen analog. So it was in kids' milk bottles, and it leaches out really, really easily. And so it started to be a very big concern that, are we actually feminizing our children by the use of this one chemical additive? Those studies were very solid and showed a very, very strong link early on before it ever got to the policy level. And that was a very strong example. So for something that's in the environment and a lot harder to track down, it's very difficult. And we'll talk a little bit later about the other thing you're bringing up too, right, is how we like to think that all science is unbiased, and it's not, right? There's politics involved in it as well. So there are some things that you can look at to try to tease apart, what you trust. Yeah. Mm-hmm. Okay. So we got projecting impact, and the other thing is anybody involved in remediation? No? Because this is another aspect of our planning and policy where the environment is concerned, that science plays a really, really big role. Now we're going not just from what is the situation now, to what is the situation coming up, but now we're saying that how can we go in and monkey with the system to get it to a state that we want to deal with. And as we go down each of these different tiers of dealing with the environment, the issues get more and more complex. Okay. So we were talking just now about perceived bias when you're dealing with scientific data, right? And everybody so far has said something about we like to deal with a scientist that we trust and that who we think is going to give us an unbiased representation of what's actually going on out there. So we know there's issues of trust and bias. What other sort of issues do you have when you're trying to incorporate science into your policy?
Preconceived notions, like if you're, a lot of what we do is somewhat trying to influence policy, providing the science to do that. But if you come into a policymaker that has a preconceived notion on something, it comes back to what you were saying, where they may have, you've got two sides of the science, and they just pick that this is the one that they like regardless
Yeah.
of what else is. So just a preconceived notion that doesn't, no matter what other science you bring forward,
I'm smiling. That seems to be a human issue. I'm smiling because a friend of mine got to an argument with her husband, and I don't know what she said, something about, you're not even thinking about what you're saying, and he said, informed or uninformed, I have a right to my opinion. Yeah. Yeah. All right, so preconceived notions, what else?
Well, interpretation of the results.
How so?
Well, like, you know, you might have two questions that look the same but are actually very different. Like on the pesticides, you know, is there pesticides in this water, or is it the compounds those pesticides turn into once it's extracted? It's a very similar question, but it's two totally different results, right?
Right, yeah. That's a really good point. So there's the finer details of anything chemical, mathematical, or health related that start getting very, very thorny. And in those cases, there's pretty much not much you could do except consult probably a lot of different experts. Yeah. What else?
Try point. Sorry.
Yeah. Oh. Yeah.
Trying to get people who aren't sciencey to understand what's actually going on. Like, I teach 10th graders. Trying to get them to understand anything can be like pulling out your hair. Like, that whole trying to actually explain it to somebody is difficult.
Right. Do you think it's a lack of interest, or do you think it's just hard and seems daunting, or, yeah?
It depends on who you're talking to, right?
Yeah. Yeah, we've done workshops for high school kids, and it can be, yeah. There's a lot of, yeah, especially right before like graduation. There's no point. Yeah, there's no point. Yeah, sorry.
Well, there's money or economics,
Yeah,
and then there's risk-benefit.
yeah. So are you talking about, see, that seems to me, or like, are you talking about when it's more of getting to be the policy side? Like trying to balance the science against that? That.
Is there any question in anybody's mind about modeling models versus empirical?
Versus empirical science?
Yes.
Okay, yeah, that is a very good question. So we're talking about how do you interpret a mathematical model, that's right, versus empirical science. That's a great point. And Matthew, I'm sure when he gets to his talk, we can talk about this some more. But does anybody have any feelings on that? I take it, do you deal with models quite a bit?
Well, I've just noticed there's a lot of difference sometimes, and the models only as good as the information that goes into that modeling.
Yeah.
Lots of times, from my own point of view, it's a little skewed what they put in. It could almost get two different, depending on what you put in.
Yep.
In Alberta, we're using, so many different types of models. I think the province of Alberta right now is actually committed to doing peer review on the modeling that they're using for anything to do with the environment right now in Alberta. So they're going to take
That would
the
be
next
great.
year, they're going to look at all their modeling for either biodiversity, water, et cetera. They're going to have that peer review to make sure that we're using the best models possible, and we have rigor in those models.
Right.
Because there has been so much of us as the public saying, we don't trust this model. Tell us
Yep.
why we're using this model when we can be using this model here. So.
That is huge. So when I started graduate school, I started in the in biosci, actually in the ecology group. And in ecology, especially a lot of the stuff that was going in that group in Ed's lab, was very mathematically driven. So it was interesting. We got to a presentation of a student who he'd built a mathematical model of something to do with populations in a pond. I don't remember. But he had an experimental system to go along with that. So what he did is he had some data from his experimental pond, built a mathematical model. Mathematical model made certain predictions which he could go back and test in the pond, right? So in my opinion, and this is a scientific opinion, a model is only good insofar as it has been tested against what has been done experimentally, which is very difficult. And when we get to climate modeling, you could have weeks of argument over something like that, right? Because there's no earth that you can monkey with to see if something's true. But what's really interesting is that even though we were a bunch of ecology graduate students, we break into discussion groups afterwards, and even the students were starting to say, I don't trust that model. You can't just model something as complex as the ecosystem. Right, so I did my whole PhD on mathematical modeling, and it is a very, very big problem. There's two things. One, first of all, you're right, a mathematical model is only good insofar as the data that goes in is good, the understanding of the processes is good, and that it's been verified against something experimentally. But besides that, there's also a inherent distrust of math as a tool, right? And that might be a separate issue. Because if physicists, everything that we know about physics, that's all mathematics. And if you talk to a physicist about building models, and they do a lot of this for biology, their feeling is, I will only trust something to be true if I can model it. Because it's going through that process that makes you be very, very specific about what you think you understand. Right? So it's a big discussion. We could do a whole nother workshop on that. Yeah. Okay.
I think there's also an issue related to time. So sometimes you don't have the results before you need to be making a policy decision, and
that's a huge issue. Yeah, so this is the way scientists think about time. I think this is a fantastic question. I think it's going to take me a year to get the system working to answer it. I think it's going to take me another 6 months to get some data from it. It might take another year or so to do the data analysis and write it up and get it out there, right? We don't think about deadlines per se, right? And from a policy perspective, it's all deadlines. I need to know before this decision goes ahead. And so I appreciate that. If you're talking to a scientist and you're like, I want to know this, they'll just go off on a crazy tangent, and maybe in a year and a half you'll get some interesting results along with about 20 other interesting results, and it's all really interesting. But there's a very different culture between what policymakers want and what scientists are able to give you. Yeah. Yeah.
The other issue that we run into a lot is data gaps. We never know if we have enough information, and can we make a policy based on what we got?
Yeah.
And we're not scientifically based, so that we don't know
Yeah.
kind of what is it baseline that we need. So that's always
That, yeah. You kind of hit on one of my biggest soapboxes. I'm just going to bring them all up here. Lack of data. And I've noticed in quite a few of the issues that I've gone into in depth, the amount of data that's lacking for Canadian ecology, I think, is pretty shocking, given that our economy is resource driven. Tell me why.
Well, because it's not in the interest of economics. And, we're obviously a province that is more interested in making money than.
Yeah. Yeah, so when I was working in British Columbia, I was working for the Forest Service. And the Forest Service in BC has its own research branch. And their job is to do any sort of research that is going to be useful for forestry policymaking. Same thing with the Ministry of Environment. Correct me if I'm wrong, but I've just been looking at websites in Alberta. Alberta Environment don't have their own researchers. Is that correct?
We do, actually.
Oh, okay, good. Tell me.
As scientists, we have limnologists that study trends in the lakes and rivers. And
Fantastic.
and write reports annually on the trends that are going on. So.
I'm so relieved I'm wrong. Yeah, that's great.
The environment's been slashed.
Also, the numbers.
Well, in the people and the work, the people in the field.
Yeah, the department over the last 15 years has had, very, very massive transformations from being quality assurance, environmental assurance, as being that sort of protected warehouse of researchers that were working on the ground, specific to policy interaction with research. Yes. They've changed the way that they do business, though, and now they work more through this partnership, and many people really do some devolving responsibility. But the whole nature of the government itself in Alberta Environment, the people in Alberta Environment, and the percentage of people doing science in Alberta Environment is very, very small compared to where it was 15 years ago.
Interesting. Okay. Yeah.
We've also traditionally, though, had the Alberta Research Council fulfilling part of that need as well.
Okay.
Specifically, they were doing work for different departments within the government. They would do
Oh, interesting. I didn't know that.
work for environment, for agriculture, for forestry. So they were all not the same.
They were also slashed 15 years ago. Steve West had one department.
Time was limited.
Mid-90s.
Interesting. And then it's a problem, right? Because from an academic scientist perspective, often what's going to get you grants isn't necessarily the very basic information that we need to know to make policy decisions too. So you'll find that scientists have a ton of pressure to maintain their research programs that will drive them away from doing things that we think as policy people would be really useful. Right. Yeah?
I think that's one that is core to all of these is the issue of funding. There's no money to do things.
Yeah. It's in part the reason that I ended up in the medical field is that there is more money for medical research. Even then, the numbers are pretty shocking. And it's a huge problem. There's also, you know, a generation of scientists who've come through the system who are looking at virtually no jobs, which is crazy considering, you know, the issues that we're facing. But yeah. Mm-hmm. Yeah.
You've got lack of data and then data overload.
Yes. So exactly. You can have either a drought or, you know, completely be overwhelmed with data, and this can happen in quite a few fields where all of a sudden it starts, you know, catching the fancy of people, and paper comes out after paper after paper. Exactly. Yeah. Exactly. And sometimes just keeping up with that amount of data is a huge issue. Yeah.
Even our means of collecting the data can outpace our ability to interpret it.
Yes.
Like I do a lot of genetic stuff, so I'll have 20,000 genes, I'm measuring gene expression, single experiment, totally overwhelmed with the amount of data that I get back. And then the tools are just starting to be put in place and the statistical techniques to handle that sort of amount of data.
Yeah. Mm-hmm. So I don't know, this is, when I was thinking of data overload, I'm thinking the same thing as you, that in the medical genetics field, it's a huge issue. Do you find that there are areas where there is just too much data for you to be incorporating into your policy, or is it always a problem of not knowing enough?
Well, I came across an article the other day about the number of drought in Alberta and
Yeah.
the prairies, and there are 1,200 papers listed
Yeah.
in this reference. Some of them could say there's no drought, some of them say it's climate induced, some people and back and forth, and take a pick and come up with an answer. And in some place in those 1,200 papers will be something quite credible.
Yeah.
And those were just the ones that had consistent nomenclature and were the sort of quality papers. 1,200.
Wow. Yeah, that's data overload for sure. Yeah.
That's actually almost more of an interpretation overload, because to be honest, most of the data
Yeah.
is based on the Alberta hydrological. It's all the same data.
Oh.
So again, that's an interpretation mode. And sometimes, when you talk about data overload, sometimes when a scientist will go in the field, you'll say, well, I want to measure the diameter, breast height of the cottonwoods on the river because that's what gives me information. Well, hell, while I'm here, I'll measure the height and all the plants that are around them and things like that. So we tend to have overload sometimes in some of the studies. We can't do anything with the data. We don't have a use for it yet, but we do have it sitting there in the background. So
Yeah.
sometimes that's sort of thrown into things as well. It's sort of muddying the water.
That's actually funny. It's a good point, and that describes my modus operandi in designing any experiment that I do. Yeah, exactly. I'll just start, and then I'll be like, I'm going to add another layer. And all of a sudden, I end up with like 60 replicates times 2, and yeah, and then you get to the end of it, and what do you do? And if we have the computational resources to be able to, systematically store all this data, it's useful, because who knows down the road that, maybe there's a hypothesis that we didn't examine at the time and can go back and look at that data. But especially when I was working, same thing with the Forest Service in BC, the data management is a huge issue. There's people dedicated just to doing that. Yeah.
And we've had examples of people or organizations that are shy now about data because, and this is not a secret, the Eastern Irrigation District for years did water quality sampling.
Yeah.
They did more than they needed, but about 5 years ago or so they quit doing it. Nobody wanted the data. So when we talk
interesting.
about a need for data and a lack of data, sometimes it's purely social and political will. If something's popular enough, if people say we need data on this, studies will start data will
Yeah.
be collected. But
Yeah.
if society hasn't valued it, then there's no funding, there's no political will, so there's no funding.
Yeah.
Everybody wants medical data because we all get sick.
Yeah. Yeah. And that's totally true, is that even for at the academic level, you know, certainly when I started this game, I thought it would be fantastic because I'll be able to choose my own questions, whatever I'm interested in, I'll be able to study. And that's not at all how it works. It turns out there are 3 major funding agencies for primary research in Canada. There's Natural Sciences and Engineering Research Council, Canadian Institutes of Health Research, and the Social Sciences and Humanities Research Council. Yeah, exactly, right? So they will set out a very large program. They say we're announcing this much in funding for this coming funding competition, and we're going to target it. So we will target our questions now towards whatever. So the latest one that has come out with CHR, we're going to target this towards personalized health, because personalized health is now something that's very interesting, and right? So now all the primary investigators who are looking at funding and gaining funding for the next 5 years are starting to think, how can I spin my research program so that it's involved in personalized health, right? So yeah, most of the big research drives, as much as we like to think that they are driven primarily by what's interesting and what's important, they're driven by the politics and by what's interesting to society as well. Yeah. Yeah.
Well, I just think that, especially in the environmental field, our needs for environmental research are long-term data sets.
Yes.
Priorities are constantly changing, and what's really required is long-term monitoring data, and that's always at what gets cut first is monitoring.
Yep.
Monitoring, and everyone wants to fund some new topic, and
Yep.
nobody wants to fund baseline monitoring, and that's what a lot of the simplistic stuff when it comes to decision making, that's the information that's required most for decision making.
Yep, I completely agree. And I also, don't you think too, that if you've started, because I remember when I was working in wildlife, you're collaring bears, right? So if you start a study where you're giving a bunch of bears radio collar, and then after 2 years you pull the plug, but there's a bunch of bears out there with radio collars, don't you think that if you've made this impact on the environment, you owe it to keep up with that data collection as well, right? But yeah, long-term studies, it's problematic for a number of reasons. It's not just the funding, it's also the people coming through, right? There's a lot of turnover in science. So, yeah. And I think this is where, at least in British Columbia, that's where the provincially based research had a lot of impact, was on these long-term studies, because they had the basis to do so. Yeah. All right, I'm probably going too long, aren't I? No one's mentioned jargon. It's got to be an issue. Yeah. But there's one thing that we haven't touched on, and that there's a really big philosophical divide between the way that science and policy look at evidence, right? So both law, policy, science, these are all evidence-based pursuits, right? We want to make decisions on the basis of the best evidence possible. And legal bases of decision-making often require a bigger burden of evidence than science can give you. So in criminal matters, this burden of proof is usually beyond a reasonable doubt. This doesn't, it's my understanding, this isn't as big an issue for environmental matters. Usually those types of decisions are made on the balance of probabilities. And this means that all things being equal, do I have 50% plus 1, more belief in something being true? If so, that's the way that the decision goes. So it's a 50-50, 51-49 split. Even in this relaxed threshold for decision making, quite often it's difficult to get the science to support that. And part of the problem is that as scientists we have a different relationship with uncertainty than a lot of other fields. Uncertainty is a huge part of what we deal with every day. Because if you're at the forefront of knowledge and you want to be investigating something, you have to be very explicit and comfortable with the level of uncertainty that you have in your ability to measure something and your ability to see an effect of something. And not only that, but there's a certain amount of uncertainty into our understanding of how a process goes. So there's a level of uncertainty associated with the data that you have, just by measurement, and then there's a level of uncertainty that goes into basically all your hypotheses. And as much as possible, as scientists, we try to be very explicit about this. And that's the reason why, as scientists will often say, that a theory or a hypothesis is never proven. We will never accept something as completely true, even if it has hundreds and hundreds and hundreds of years of support for it, unless you corner me in a bar and ask me about evolution, and I will tell you it's 100% true. But in general, the idea is that we never accept something as completely true. And that all of our experiments are designed actually to disprove a hypothesis no matter how dear it is to you. So that kind of caution, when you're talking to a scientist, is often really hard to communicate. So I had a colleague of mine, she did this fantastic course, and it was on science literacy. And so they had a group of science students and a group of public relations students and a group of journalism students. And they put them in a room. And then they got them into groups, and they said, okay, the scientist is going to tell the PR person what they work on. The PR person is going to tell the journalist what the story is. And then we'll get the story out and see how it goes. So my friend Anne, she was working on salmonella. And so she told the PR person about, I work on salmonella and signaling between, the different cells, and, this is why it's interesting. She gets the article back from the journalism student, and now it's gone from she's working on salmonella, and her research is going to cure the common cold and cure AIDS and fatal halitosis. Not making that up, fatal halitosis. It's bad breath that'll kill you. It was like the worst game of telephone ever. And it was really heartbreaking for Anne, and you actually feel like a sense of panic anytime that something you've said has been what you consider to be overblown, because that sense of uncertainty and caution is so ingrained. And so that's why often when you're speaking with scientists about, the research that's going on, there's a lot of, well, the evidence would suggest, or, so far it looks like this. But communicating that uncertainty is a huge issue. And so something like, did anybody read the International Panel on Climate Change report on uncertainty? They had a bunch of recommendations. They're just fascinating. And the recommendation for the third report was to have all of the authors be very, very explicit about uncertainty of all the primary literature that they came up with. Because when we're going into, a bunch of literature, and we have 1,200 papers to read through, right? We want to look at what does the study tell me and take the main point from it and then write it down and then move on. And that's how I work. I'll be honest, right? Ideally, we'd also be able to say, what is the uncertainty associated with that estimate? Because if we're building up a whole bunch of them, right, you want to be able to know, A, what you can trust, and B, how that uncertainty is building or not building into your answer.
Who determines the uncertainty?
Ideally, it's actually a mathematical issue. It really is something that you can measure. Yeah, it should be very statistical. So in the case of the IPCC, these are numbers that start accumulating. Yeah, and there's a great figure in it.
But somebody designed the model in order to come up with those numbers.
So there is a way to proof a model that involves taking every parameter that feeds into it and varying it as much as is reasonably possible, right? Like this number can vary between 0 and 500. So now, exactly, right? So I'll put all of those numbers into the model. Let's see what the range of numbers that I get out is. Let's see how reasonable those are.
This is just tearing, because once you get too many parameters, you can't do that anymore.
Yeah, it's true. I tried to do that for a model once, and I wrote a computer program to do just this to a mathematical model. Hit go and waited and waited and waited and waited and waited, and I realized that in order for it to do this one process, it would have taken 3 years on a desktop computer.
And that's only if you've identified all the variables.
Yeah, exactly, yeah. And quite often models have, there's a branch called partially specified models, where if you don't really know how something works, but you think it takes this shape of this line. So I think that, let's say, snow melt's going to go up with temperature and it'll be a straight line. It's called a partially specified model. You can just put in a function that you think is going to represent what's going on. So even, there's not just the actual parameters sometimes that we're unsure about, but these actual processes. This is interesting. I didn't think that, modeling would end up being such an issue, but this looks like something that we could probably spend a lot of time on. Yeah. Okay. Where am I? Okay, so then we're all here because we're like, what are we going to do? And so part of the problem, I think, is really just increasing the conversation between primary science and policy. And I found this joke in a government document, which I thought was just brilliant. But a man in a hot air balloon is floating along, and he gets completely lost. So he sees a break in the clouds, and he comes down, and he sees a man, and he decides to ask for directions. So the man tells him, you're at 45 degrees, 25 minutes, 29 seconds north, and you're at 75 degrees, 42 minutes, 20 seconds west. I'm at about 100 meters above sea level, so you must be at about 120 meters above sea level. And the balloonist is just beside himself, and he says, you got to be a scientist. I ask you a simple question, you give me too much information, and I'm still lost. And the scientist, or the man on the ground says back to the man in the balloon, you've got to be a policy analyst. You came out of nowhere with your questions. I give you the most accurate and precise answer that I can. You're still lost, and you blame me. I thought it was funny, because honestly, I'm a scientist, and I started reading this, and I'm like, what is a balloonist doing without a compass and a GPS? Like, that should be perfectly useful. Yeah. So, anyway.
That's a great answer.
Yeah, exactly. So this is where we get to the tools part. Now I'm going to start giving you some very specific tools so that when you go to some primary literature, you're going to be able to apply a set of criteria for how well do you think this study is done. Obviously, there's some statistical, mathematical, chemical, like some very specific issues that you have to deal with in your own realms of inquiry that are too specific to get into. But these are the very basic fundamentals of science that should ideally frame everything that gets published, and it totally doesn't. But this is the ideal situation. So let's just start out with a discussion. So can anybody tell me, what is a hypothesis? Exactly, right. So if you say something as an if-then, this is a question that can be tested. If I think that something's happening and I do this, then this should happen. So is there, like, we can make an example out of this. So somebody give me a question that you're interested in with your own research. How about, do pesticides have an impact on human health? Right? Okay, so how would we frame a hypothesis out of this question?
If you consume however much of whatever, you will get whatever.
I love it. Exactly, right? So that is a hypothesis that's so specific that it tells you how to do the study, right? So now you get a bunch of people to eat some pesticides, right? And you measure them for whatever disease you think it's going to give them, right? So obviously that's not always, it's not going to get you path ethics, I'll tell you that, right? But it's a great hypothesis, so, right? So a really good hypothesis is going to frame how you do the study. And even when we're doing ecological research where, you know, this kind of direct experimentation is really difficult, something like if exposure to chemicals or to pesticides causes X, then we should be able to see it this way. People who live in a municipality that uses a lot of pesticides should have a higher rate than people who live in a municipality that does not. Something like that. Make sense? Okay. Can anybody tell me what a control is? Yeah, exactly. So?
When you do your very best to keep the same.
Yeah, exactly. So it is a section, the group of replicates that you don't apply the treatment to. So then you have a way of comparing things, right? So there's lots of different things to think about, though, when we're using controls or setting up controls for an experiment. And I'll tell you that when it gets to publication, the majority of papers that I've had that have been rejected and the majority of papers that I've rejected are due to improper controls. It is very thorny because not only is there the main thing that we're looking for, but there could be many, many underlying processes that may be giving us the results that we're looking at that we haven't thought about. Right? So what would be a good example of something underlying that will mess with your results? Say.
Existing condition that you know about or
Yeah, like a pre-existing health condition. Is that what you mean?
I was thinking environmental.
Oh, okay, sorry.
Same thing.
No, a pre-existing environmental condition. Yeah, exactly. Or so, what about something like the geography of your study sites? Right, so a lot of these studies take place over very, very large areas. They're often north-south gradients, altitude gradients, and temperature, and humidity, a whole bunch of different things. And these are things you have to consider. Time. Time is a huge issue. It's a huge issue statistically. And this is something that doesn't get picked up on a lot, but it's something to be really, really careful about. Because when you're doing your statistics, almost all statistics assume that every number you give it is independent of the next number that you give it, right? But let's say, for example, you're counting the number of fish in a pond, and you count every single fish in the pond. And you do this count every day of the week, and now you start comparing numbers. Do you think that the number that counts from Monday and Tuesday, are they independent? Is there any reason to think that those numbers will be similar to each other?
Well, it depends if the fish are migrating up and downstream or something, they might be on their way through.
Right.
So there would be, the same as geography, there's that spatial autocorrelation.
Exactly, right? Yeah, there could, definitely be. Now you're into really complicated mathematics if you have space and time interacting, for sure. But let's say, in that case, what if you do that count 5 minutes apart? Do you think that those are really independent measures of what the number of fish is? No? How come?
They're not, I wouldn't say they're independent replications. I would say that you're repeating the same thing twice. It's not.
That's exactly it. Yeah. You're counting the same animals the exact same time. So it isn't actually an independent replicate of your count, right? It's very dependent on what happened 5 minutes before. So when you see anything like a long-term temperature trends, there's a high degree of autocorrelation in those temperature trends. It's something to be aware of. It doesn't mean that that data has to be thrown out, but there is a branch of statistics that will be able to handle anything that has a level of autocorrelation with it. But it's something to be very aware of, because I've seen it time and time again, data that is actually autocorrelated due to time or space and is treated as though it's independent.
I'm sorry, I
Yeah.
don't quite get that.
Okay.
So if you go back to your fish sample and
Right.
you're counting on Monday, not counting on Tuesday, you're saying that those are not independent?
No, so think of it this way. I have counts Monday, like every day of the week. What is more likely to be different, Monday and Tuesday's count or Monday and Thursday's count? If you,
Never include, do you?
well, let's say if there's birth, death processes going on, right? So the chances of there being a birth or death in a 3-day span is higher than in a 1-day span, right? So that, yeah. Okay, does that make more sense?
Yes.
Okay, good. Seasonality is a really big one, and that'll come up in one of the exercises, hint, hint. And then another big one, and this is the thing that we're always on the watch out for, is experimental artifacts or mathematical artifacts. And it's pretty easy, actually, to introduce an artifact into your experimental design by using the wrong filter, by using the wrong buffer, by doing the wrong transformation of your data. And in fact, in my big modeling study when I was in grad school, I had introduced a mathematical artifact into my calculations that turned one number from a positive to a negative. And it took me a year to find it. It can be very tricky. Yeah. Oh, like physical ones. So like if you're doing some sort of chemistry, a buffer is just the stuff you put it in so nothing changes by the time you get it to the lab. Yeah. Sorry.
So for the non-scientific artifact,
Yeah.
it's just an element?
Or it changes something that you don't think about, right? So as an example, right, I'm just doing a bunch of calculations, something turns negative, all of a sudden I get a bunch of negative answers. And I think, wow, I have negative regulation here. There's a feedback that I didn't consider. No, that's not what happened at all. It's just because you set up your equation wrong. It's like a mistake, essentially. Yeah.
And when I worked in a nutrition lab in
Yeah.
university, and we were using plastic pipettes, and then we switched to glass pipettes, and we started to get completely different results.
Really?
So it was like even just the actual scientific stuff that you're using can be an artifact.
Wow.
3 terrible months.
It's true. I mean, it happens all the time, right? And then you get months and months in and realize. And yeah, actually, same thing. I'm in the middle of an experiment right now, which would work if I was in just regular salt water as a neutral buffer, right? Everything's working fine. And then we moved it all into, when you get medical samples, they come in a different buffer, and it stops working. We've been monkeying with it for a year now. And it turns out it might be because there's magnesium in the second buffer. That's a year, you know, but that's how easily these things can creep into an experiment. And so when you're reviewing primary literature, it's very difficult unless you're in the trenches to know where to look for these things, and there's nothing really that you can do to try to tease that out. But what you hope is that peer review has caught it, and that's the role of peer review, is to catch these things, and we're going to come to that in a minute. Okay. All right, so controls, very big issue. Replication. I just got it, yeah. Okay, replication. Does anybody tell me what replication is and why it's important? I think we've kind of touched on it a little. If you did an experiment once and you're like, awesome, I have negative whatever, are you going to go and publish it? Are you going to take it to your manager and say, this is what happened? No? How come? Yeah, exactly. So replication is exactly what it's for, is just to make sure that you're getting the right answer. And in a statistical sense, what it's doing is increasing your power to be able to detect an effect. But there's a trade-off. There's a huge trade-off, and this is a very, very big issue when you're doing ecological studies, is often there's a very, very big cost to doing this experiment over and over again. So that is a very big balance. There's a cost in time and money versus the power that you have to detect what you're looking for. If I say that an experiment is fully randomized and blocked, I'm sure that we've heard this in the media and talking about medical experiments. Does anybody know, can anybody explain to me what that means?
You mean a randomized complete block design?
Yes.
So you mean, the only perspective I come from is from an agriculture one when you're doing field studies.
Yep.
When if you have a row of, say, different beans, you've got A, B, C, and D. The next row, you don't want A, B, C, D, A, B, C, D, A, B, C, D, because that's not a random complete block design, because there might be some effect, say, on the left side of the field that's always only affecting A's, or on the right that affecting D. So ideally, you want a computer to randomize it for you.
Yeah.
So that you might end up with an A, B, C, D, D, C, B, A, whatever, to take out some of the underlying parameters.
Exactly. That's exactly it. So randomization means you give your replicates to whatever treatment in as random a manner as you can, and blocked means that they all get the same number. So it's actually quite simple. Every treatment gets the same number of replicates, and they are assigned randomly. Okay?
The problem in policy, though, is often if you get the answer you want, you go with it.
Yeah.
That's the problem.
It is, and it's not just a problem in policy, it's a problem in science. Yeah, it really is. Yeah. And I think that's where our responsibilities as, consumers of science comes, right? Is that we should ideally be able to at least have some skills to look at these and be able to pick it apart and say, I don't get it. I don't believe that. Okay, and so peer review. This is my favorite part of the process. So can anybody explain to me peer review? Because it's people who are involved in the field, we'll look at everything like, have you introduced mathematical artifacts, experimental artifacts, controls, the interpretation, the statistics, the hypotheses. All of these issues that we've looked at, as well as the nitty-gritties, will be subject to review. And so a paper after this review can be rejected by the journal outright. The journal can say this isn't suitable for publication. The journal can come back and say, kind of like where you're going with it, we suggest these other experiments that the reviewers have suggested. We suggest you do them and invite you to resubmit. The paper or the journal can say, we will accept your paper with major revisions, and that's usually things like interpretation of the results or statistics. Or the journal will accept the paper with minor revisions, which may be, you missed a reference or you spelled something wrong. In my experience, almost no paper gets accepted the first try. So this process can take months, and this is a big issue when it comes to policy decision. It's part of the reason that things take as long as they do. Yeah?
So without sort of repeating it, if you've already answered it.
Yeah, no, please.
Ever did
is a very, very good question. We were talking about that this morning, actually, some people that I work with. Because the paper itself didn't make any of these big claims, it was very innocuous. All it really said is that these kids have a bunch of GI issues, and there's, you know, they happen to get the MMR vaccine, right? They didn't go so far as to say that there was a link. It was only at the press conference afterwards that Wakefield said that there was a link. I don't know.
Study of 12. I mean,
In medical, in case literature, that's not that uncommon. Like a medical journal will often write, you know, I have a case report of one patient who came in with something interesting. Yeah, it's difficult, and it's a judgment call. Yeah.
Can you talk about stratified versus blocked?
You're testing me.
I just thought of it.
So stratification, he's asking the difference between stratifying and blocking. So stratifying is actually a form of blocking, where you are, you're more explicit about what those blocks are. And so actually you can be dividing something into, the geographic gradient is a good example of this, right? So I have a north-south gradient, I'm gonna stratify it, say, every 5 degrees of latitude or something like that. Does that answer your question?
Yeah.
Okay. Anything else? All right, so I've given you guys some tools, and we're gonna do a quick test of looking at a study and seeing how we feel about it. All right. We're gonna start talking about cats. So in New York City, there's a lot of high-rises. There's a lot of cats falling out of high-rise windows. It's a huge problem. It's actually called high-rise syndrome. It's a suite of injuries that cats get from falling out of high-rises. So there's a veterinarian in New York City who started noticing something. He noticed that, you know, depending on what floor, and I should start this off by saying this isn't actually the graph from the original paper because it's really hard to find. It's from 1984. This is data that I've completely made up for the purposes of illustrating a point. So I should have my name attached to this. Completely fabricated. Okay, so depending on the number of floors that a cat falls from, he starts getting more and more injuries until you get to the 6th floor. And something interesting happens. Cats that fall from higher heights start getting fewer injuries. And I thought, this is really interesting. Do you know the punchline? If you know the punchline, hold it in. We compare it to this if it happens to humans. Again, it's completely fabricated. Humans just keep getting worse and worse hurt no matter how high they fall from. Right? So, so the vet thinks this is really, really interesting, and he starts going back into the admittance records of a bunch of cats that have come into his clinic with high-rise syndrome. And this is, you know, he generates all this data, and he finds, in fact, that this is true. He does see this association that, you know, cats who've been admitted to his vet hospital with this number of injuries, and this, anyway, it all follows out. So this story first came out in 1984, and it was actually a pretty big story, and it keeps cropping up, and people are like, well, why aren't these cats getting more hurt? Maybe after 6 stories, they hit terminal velocity, and they can right themselves, and then they're relaxed. And so when you hit the ground relaxed, maybe you get fewer injuries, and humans don't know how to right themselves, they don't know how to relax. So these are all possibilities. And I just love this story. It always cracks me up. National Geographic, 2 years ago, put up a video of this story. So this is still floating around out there. And Lisa, do we have the video? That's okay, we're running short of time, I'll just skip over it. So I've given you the basic story, and National Geographic still thinks it's really interesting. So now that I've given you the tools, can anybody tell me what's wrong with this study? Yeah?
It doesn't tell you how many died.
Ah, exactly. The dead cats don't go to the vet. That's exactly it. If it's a dead human, it goes to the hospital and there's a record. This dead cat, you're gonna get a shovel, and no authority is ever gonna hear about it, right? But this is something that made it into a journal and got to be a pretty big story. Yeah, and National Geographic's still talking about it. I'd like to introduce Matthew Morris. He's a PhD student from the University of Calgary. Super interesting guy if you get a chance to talk to him. And he's done work on the water ecology kind of group at the University of Calgary, and he's going to be talking about anthropogenically driven evolution. And this is your master's research, is that right?
What I'm presenting today?
Yeah.
No, it's
Okay, never mind
tangential. My master's research, I was afraid none of you would be interested in it.
Okay.
So this is work that is related to my master's, but it's not going to be my work, which for me, now that we're also very skeptical about the scientific process, is better, because now we can tear this apart, and I won't feel awful
And
at the end of the day.
no hurt feelings. Okay, that's good. Way to go.
But I did do my undergrad with Dr. Jeffrey Hutchings, who is one of Canada's leading experts on fisheries-driven evolution. So my particular honors work with him involved aquaculture, farmed wild salmon and their interactions, which was one of the things I was thinking about talking about, but fisheries pressure, I think, is also remarkably interesting, and I absorbed a lot of this information when I was working with Dr. Hutchings. My goal in the next 20 minutes or so is simply to convince you as policymakers that evolution is important, that it is something that we should be taking into consideration. The work that I'm going to present isn't necessarily all perfect. I'm sure there are things that we could rip up with it, but I'm going to present so much work that you couldn't possibly doubt at least the general message. But we'll go back in time to start, go back to the 1840s, 1850s. Charles Darwin had a remarkable appreciation for the capacity of humans to alter the organisms that are around them. What you're looking at here is a diagram from his book Variation Under Domestication, which came out somewhat after The Origin of Species. What you see here are a variety of birds. In the middle we have the rock pigeon. This is the standard pigeon that we're all quite familiar with its sort of bluish back and the banding on its tail. These are the things that are the bane of city dwellers the world over. And you see all these bizarre sorts of birds. This is a fantail, which looks kind of like a peacock in its tail feathers. We have the pouter with this huge breast, the carrier with a really long, skinny neck and this funny-looking beak. And what Darwin appreciated was that all of these birds, if you were to take their skeletons, take them to an ornithologist, they would say these are different species, probably in different genre, if not different families. But in fact, all of these are breeds of the rock pigeon. Every single one of them has descended from the rock pigeon. And we know this because we're the ones who did it. Humans artificially selected certain pigeon varieties that descended from this rock pigeon to produce, over time, these very bizarre looking creatures that had never before been seen in nature. Now today, you and I would probably be more impressed if we looked at dogs, like the St. Bernard and the Chihuahua, how genetic studies have shown that they all descend from the gray wolf through thousands of years of domestication. Or we can just go to the grocery store. You go to the grocery store today and look at the vegetable section. You'll see broccoli, cauliflower, Brussels sprouts, cabbage, all in different places at the grocery store with different labels, different prices. But in reality, they're all the same plant. They've all descended from wild cabbage. From this weed-looking organism, farmers have selected variants to produce this abundance of different crops, but they're all the same species. This is just the power that we humans have over nature to shape and alter it for our own particular purposes. Now for Darwin, evolution was this very slow, gradual process that took extraordinary lengths of time. He went so far as to look up Roman records to show that the ancient Romans had domesticated pigeons, just to show everybody that thousands of years are required to produce these pigeon breeds. And our policies tend to follow this assumption. We tend to base policies on ecological considerations because the ecology changes quite rapidly in our own lifetime, right? Like we can see how interactions between organisms are altered. We can see habitat degradation, population size, all these sorts of things. But generally, historically at least, our policies were not based on evolutionary considerations because what is 50 or 70 years of a policy? It's just a blip in the evolutionary timescale. Our recent work over the last 2 decades has shown that evolution can be very rapid, and that humans have the capacity to not only alter domestic organisms, but we can alter wild organisms without even intending to, and we can alter their very genetic structure. Now, of course, the first thing, and this is one of the problems when we talk about science, is these words that we use and what do they mean, because different people have different definitions of things, and evolution is certainly a word that different people understand in different ways. So I thought it'd be best to give a baseline definition of evolution. And first I'll talk about what evolution is not. So let's assume we have a population. These are all different individuals, and these individuals vary in the color of their tongue, Some have a blue tongue, some have an orange tongue, and we sample the population and find half of them are orange, half of them are blue. But as we investigate, we discover this has nothing to do with any sort of genetic basis. It's purely environmental, in that there are popsicles distributed throughout the habitat, orange and blue, and whatever popsicle the organism first encounters, it licks it, and that becomes the color of that creature's tongue. So we go back 10 generations in the future, and we discover, oh, the colors switched on me here. But let's assume that this is blue, and this is orange, so there's now, well, just ignore that. There's a greater proportion of orange individuals in this population now, so something's changed, but we would not say that evolution has occurred, because nothing here is heritable. The next generation, these orange individuals will not be passing on orange tongues to their offspring. So instead we would wonder what's changed in the environment. Perhaps there's been some great blue popsicle melt that occurred, and the orange popsicles remained, right? So there's environmental change that has caused phenotypic change in the population, but none of this is evolution. If we swap, however, popsicles for genes, let's say that there is a gene that controls tongue coloration, and we have 50% orange, 50% blue, and let's say that there's a variation of this gene such that individuals with allele A have a blue tongue, with allele B, you have an orange tongue. Now, an allele is simply a variant of a gene. Okay, so you have one gene, you can have multiple types of that gene, and in this case, allele A gives you your blue tongue, allele B gives you an orange tongue. Okay? So then we sample 10 generations later, and we see that the population is now dominated by this orange tongue. So allele A has come to dominate the population, or sorry, allele B has come to dominate in the population. We have to wonder why, what's causing this change, and we would say, irrespective of knowing the cause, that evolution has occurred. The frequency of the allele has changed over time in this population. Whether it's due to chance or it's due to selection, it's still considered to be evolution. We'd have a more complex scenario where we have 10% of the population in generation 10 is something completely new. This black tongue caused by allele C, and if we could go back, say, to generation 5 and see that a migrant came into that population and has spread that allele throughout, we would say that evolution has occurred because it's heritable and it's been passed on to another generation. So I'm defining evolution in this case as changes in allele frequency over time. So I'm not talking about, birds or lizards sprouting wings and flying, or fish getting legs and walking on the ground. This is simply changes in allele frequency over time, which is a really boring and not very exciting definition of evolution. When it comes to determining whether evolution is happening in our lifetime, this is certainly a great definition to have. So this evolution is fueled by genetic variation. In this example, the variation has always existed, but the frequencies are changing. In this example, there's some sort of new variant through a migrant or a mutation that's starting to spread throughout the population. But it's this genetic variation that fuels evolution, and then it can be driven by something like natural selection, where one particular allele has some sort of benefit over another allele, so it comes to dominate, or something like what we call random genetic drift. So this is just random changes in allele frequencies, usually in small populations, where one allele might get lost and the other might be favored, totally due to chance, has nothing to do with the value of that particular allele. But the key point here is that without variation, there can be no evolution, and therefore no adaptation. So the question that I wanted to ask is, can humans unintentionally drive the evolution of wild organisms? We certainly can do it intentionally. We have with our pigeons and our broccoli and our dogs, but can we do this without even trying? So the example I want to talk about is fishing pressure, or just the fisheries in general. And a question that has been asked and debated for several generations now is whether the fishery is like a lawnmower or like a sieve. Now, if the fishery is like a lawnmower, you go out with your lawnmower and you cut the grass, you trim it down, and then the grass grows back, and the lawn after it's grown back is the same as the lawn before you cut it down in the first place. So is the fishery, we remove the fish, and then the fish rebounds, we've taken some maximum sustainable yield, the fish responds by producing more fish, the population after it's responded is the same as before the fishery, or is it more like a sieve where those nets are coming in and removing particular alleles from the population, sparing other alleles, such that those alleles come to dominate in the future? Well, before we can talk about the fisheries, let's talk a little bit about hunting and some of the effects that hunting can have, because it's way easier to study land animals than it is to study fish, and maybe you all appreciate how hard it is to study land animals, so it's remarkably difficult to study fish. It got cut off at the bottom, but this is a paper from Coltman, U of A, looking at bighorn sheep from the 1970s to 2005. This is a particular population where you saw the average weight of individuals was declining over time, the horn length of individuals was declining over time, and this had nothing to do with population size, because the population had been fluctuating during this time period. Now, the conclusions that they made from this was that human predators are different from wild predators. So you very rarely see a hunter with a bighorn sheep that has tiny horns, it's missing an eye, it's mangy, and it doesn't have a leg, right? Now, an animal like that would be a prime target for a mountain lion, but as humans, we want the strong, the virile, we want the trophies, right? So these trophy hunters, the story goes, are coming in, removing these beautiful male bighorn sheep, and it's these males with the largest horns that normally contribute the most to the next generation. These bighorn males have access to more females, they'll have a harem. The smaller horned ones don't breed or breed very rarely, so the bighorn alleles get passed on to the next generation. But we kill these individuals before they've had a chance to breed, or before they've had a chance to meet their full reproductive potential. Meaning that now the smaller horned individuals have a better chance of breeding, they pass on those alleles to the next generation, and now we have a population dominated by small horned bighorns instead of big horned bighorns. If that makes sense. Basically, there becomes a survival advantage to not being a trophy, because as humans, we want the trophies. Now, a meta-analysis by Enviornmental et al. In 2009, they wanted to know, okay, so we have all these little isolated examples, but how does that rate of evolution and these hunted systems compare to natural systems? So we looked at 40 human-harvested systems. These included plants, fish, and large mammals. And they compared the rate of evolution in these systems to the rate of evolution in naturally evolving systems. These are things like Darwin's finches that we've been following for generations. And then systems that humans have impacted, but we haven't been harvesting those organisms. So, for instance, if we moved a fish from one lake to a completely different lake and have been tracking changes over time in those populations. And what he found is that on average, human-harvested evolution was 3 times more rapid than what existed in nature, and was 50% faster than in systems impacted by humans. So their conclusion here was that us humans, as these unique predators, can actually speed up the rate of evolution above background levels, above what we normally see in a natural system. And they found that, for instance, body or horn size declined in 95% of the cases with an average change of 18%. So on average, say your horn size would have decreased by 18% from before the hunt. Or things like life history traits. So the age at which you become reproductive, or the size at which you become reproductive, was also declining in 97% of cases with an average change of 25%. So if you were breeding at the age of 4, now you're breeding at the age of 3 on average. One particularly interesting example involved elephants in Africa. In the 1930s, on a particular game reserve, 1 out of every 100 male African elephants were born tuskless. This is just a genetic variant that these particular elephants happen to have. By the 1980s, it was 1 in every 10 males were born tuskless. The idea being that poachers value ivory, they will kill those males that have ivory and will spare the ones that don't. So you could imagine 200 years in the future, if this continues, elephants might still be around, but ivory might have gone extinct because we've removed the alleles that produce that ivory in those males. Now when we get into the fishery, we've seen some of these same trends where individuals are on average breeding earlier and at a smaller size than they did before the fishery, and we have data for cod, salmon, whitefish, plaice, fish, things like this. And this isn't necessarily a great thing for these, for the recovery of these stocks, because fish that breed at smaller size produce fewer eggs. So if you're maturing at the age of 2 instead of the age of 4, you're not going to be producing as many eggs when you reproduce. But there's this tremendous question, is what is causing this? Is it environmental change, or is it evolutionary change? So let's say this is what we observe. We observe before the fishery, there's all these large fish that are reproducing at a large size. all these fish reproducing at a small size, they're all mixing together, you know, possibly due to different alleles, but maybe not. And after the fishery, we see a predominance of fish reproducing at a small size. Now, if this is evolution, the scenario you could come up with would be something like this. We preferentially remove the large fish. You know, we want large fish to eat. They look better on the table. Our nets have holes in them that the small fish maybe can wiggle out of while the large fish get trapped. So the small fish contribute more to the next generation, passing on their alleles such that we get this observed increase in small size. That's one possible explanation. And if this is the true explanation, then it means that fish recovery, to bring them back to this sort of state, is going to take a considerable amount of time if we haven't already removed all the large size alleles entirely. Now, if it's environmental, you could imagine a situation in which, let's say, these stars are food. Pre-fishery, there's a certain amount of food, but there's a lot of competition for that food. We remove a bunch of fish. In this case, I'm just showing equal amounts of large and small. So the amount of food can increase because there's fewer fish eating it, which means there's less competition. So there's the fish get more food per fish than they did before. Now, when fish eat a lot, they grow faster. When fish grow faster, they tend to reproduce at a smaller size and at an earlier age because they have the energy to invest in reproduction at that time. So the environment could be causing these sorts of scenarios. So this leads to a really difficult conundrum, because if it's the environment, then you would expect once the population recovers, you'll end up with the larger sizes again, because evolution hasn't occurred. This is just solely environmental change. So when it comes to policy decisions we want to make governing the fishery, we need to know, is this the environment or is there something genetic going on behind the scenes? So this is an experiment from Conover and Munch in 2002, published in the journal Science, which has now taken a big beating today, so you guys won't trust anything from this paper. But they wanted to do some experimental evolution to try and see if they could alter the size and growth rates of fish and whether these changes would be heritable or not. So they would control for the environment and see if evolution occurred. So they had 6 captive populations that they formed from this huge gene pool, 1,100 juveniles per population, so these were pretty large sizes. They controlled for density by having the same number of fish per tank. They controlled for food by making sure there was plenty of excess food, so there was little competition, so all the fish were growing as fast as they could under that given density. And they had 3 treatments with 2 replicants each. So those 6 populations, 2 of them went to the one treatment, 2 to the other, and 2 to the third treatment. So they were essentially doing this experiment twice, but at the same time. The first treatment, they kept the 10% of the fish that were the largest. So they weighed and measured all the fish, and then the upper 10% they kept, the other 90% they discarded. So they're having a huge fishing mortality in this experiment. The second treatment, they kept the 10% smallest, and the third treatment, they kept a random. Whether you were big or small, they just randomly took 10% of the fish and kept them. Then allowed the survivors to spawn, and those offspring they weighed and measured to produce the average weight and growth rate for those fish. So they didn't use the ones that they were keeping, they used all of the fish each generation, and then they repeated by removing the upper 10% or the lower 10%, keeping those ones, removing the 90%, sorry. And they did that for 4 generations, okay? Does this make sense, what they did? So you got a nice control here, in that you're taking, you're removing 90% randomly and keeping 10%, so they got nice controls, they got some nice replication. You may question the choice of 90% mortality, which is tremendously huge. When we're doing experiments and we're working with fish, a vertebrate that reproduces once a year, you don't have that much time to do an evolutionary experiment, but it will call some stuff into question. So what they found here is the control, which they centered on 0. This is growth rate, millimeters per day. So that's centered on 0, so you can see the differences between the other 2 treatments. And what they found was that the parental generations' growth rates were the same for all the fish. Then when they kept the largest 10% and allowed them to spawn and looked at their young, they grew a little bit faster. Then when they took the 10% of those, they grew about the same as before, but over time there was a general increase in the growth rate of this population. And for the small fish, there was a general decrease in the growth rate of that population, which indicates that there's something genetic going on, there's something heritable. The environment is not causing this change. There are alleles that are being removed, and each treatment such that the averages are shifting. Make sense? They also looked at something like the average weight of a harvested fish. You can see the controls fluctuated a little bit over time, but it essentially stayed the same as it was at the beginning of the experiment, about 3.5 grams. When they kept the largest 10%, by the end, there was about a 1 gram shift in average weight. When they kept the smallest fish, there was about a 1 gram decrease in average weight for these fish populations. And again, this is not measuring the 10% of the smallest, this is measuring all of their offspring. So they concluded that evolution has indeed occurred for the average weight of these harvested fish. And again, they looked at size and age for these fish. So at day 185 in generation 4, they looked at the average weight. And here you're looking now at all the replicates. So here are the 2 replicates for the large capped, for the random capped, for the small capped. And what they found is that at day 185, when they kept the large fish, those large fish were on average larger than the small fish at the same age. Now what they didn't look at was maturity, which is a real shame for this experiment, but at least they showed that size is heritable, and that size can evolve rapidly. 4 generations of very strong selection. So they concluded that there was indeed evidence for fishery-induced evolution. By removing the alleles that make a fish large, they're favoring the alleles that make a fish small, so those alleles increase in the population, and the reverse is also true. And this leads to an open question, however, to what extent is this reflected in nature? As I said, this is a 90% fishing mortality, which is huge. And these are for fish that reproduce every year. What about for salmon that reproduce every 4 years with a fishing mortality that maybe is 30%? How rapidly would evolution be occurring in a system like that? And that's where it becomes a much more open question, and again, is really difficult to determine, which is why these sorts of experimental or these evolutionary experiments are so important, because at least they show us that it's a possibility, that even if you control for environmental effects, you can have these changes. So it means in the wild, there's at least the possibility that this is what we're seeing, or a combination of environmental and evolutionary effects. Now you can ask, why is this important? Well, fisheries-induced evolution can be harmful to us if it's decreasing our maximum sustainable yield. If by removing large fish, we're favoring small fish, we need more fish to produce the same sort of biomass. Perhaps the variability that we're removing, if we're removing these large alleles, perhaps those alleles are important to meet environmental challenges that haven't occurred yet. So if we remove them, we reduce the ability of that population to evolve and respond in the future. And finally, it might, for the same similar reasons, reduce their ability to recover after a collapse. Now we've discovered this in the fisheries and in hunting. I could also talk about aquaculture and how there's recent evidence to suggest that within 6 years of domestication, or 6 generations, sorry, of domestication, farm salmon are genetically distinct from the wild salmon from their source river. So quite rapidly we can alter the genetic structure of aquaculture production. So, but really, who cares? What does it matter? It's becoming increasingly clear that our policies can influence the evolution of organisms. So our fishing policies in the past have tended to be keep the large, save the small. Well, that might not be the best strategy if you want to maintain a large size in the population. A better strategy might be remove some of the small, remove some of the large, save the middle, or perhaps throw back half of your large and half of your small and half of your middle to preserve those sorts of alleles in the population. But at any rate, it seems like we might be removing alleles that we need commercially. You know, a hunter's not going to pay 10 or 20 or I read somewhere $100,000 to hunt bighorn sheep in the mountains if those sheep don't have nice, beautiful big horns. And again, reducing genetic diversity and shifting population averages. But this is where, it becomes an open question for you guys, because as a scientist, if farm salmon are escaping and interbreeding with wild salmon, I can't tell us whether we should care about those sorts of things. I can just tell us that it's happening and what happens when it happens. Or if we're going out with these fisheries and removing these large fish, I can't tell you that it's the most horrendous thing in the world that's happening. I can just tell you that it is happening and that maybe we need to do something about it. But really, what is our ethical and legal responsibility for these sorts of things? currently we preserve evolutionarily significant units, but should we be preserving the individual genotypes within those units to maintain that genetic diversity? And to what extent do our decisions affect the evolution of poorly studied systems? So we know for fisheries, but what about, say, toxic concentrations that we allow into our rivers? We talk about things like LD50s, where 50% of the organisms die when they encounter a particular concentration of toxin. Well, that means there's probably a genetic basis for why 50% of them are dying. So are we shifting it so that now we're making a 50% that are immune, or favoring those alleles that are immune to that sort of toxin? How exactly are we causing them to evolve? And in those ways, it's remarkably understudied. So that's all I got to say in that short little bit of time, and hopefully we still have time left for the next thing, but thank you, and if we have questions, I'm more than welcome or willing to talk about it.
Does anybody have any questions?
I have a question. If you have 6 or 7 different salmon farms, would they each, after 6 generations, have 6 or 7 different genetic?
That's a fantastic question. And if we had time, I was going to talk a little bit about it. Roberge did a paper in 2006 where they took wild salmon from the St. John River in New Brunswick. That is the river where all of our farm salmon in eastern North America come from that river. So they took wild salmon from that river, and they took the farm salmon that had been in that river 6 generations ago, and they looked in. Okay, there's like, they looked at gene expression, and about 1.5% of the genes they looked at showed differences in terms of gene expression between those groups. They then did the same thing in Norway. So now we're talking about a Norwegian river, and then salmon that had descended from that river in the farmed environment. And again, there were genes that were differentially expressed, but 16% of those differentially expressed genes were the same between the 2 farmed populations. So they said that this is an example of parallel evolution, that there are particular genes that are being selected in both farmed environments, even though these are fish adapted to different environments, coming from different geographic regions. But there was some similarity in selection in those environments. So in Canada, you can imagine the same sort of thing, that there's likely the different salmon farms, there's going to be some differences due to drift, but it's going to also depend on the policies for how they interbreed between the different farmed environments and things like this, but there will be some genes that are certainly similar.
I mean, you could say that evolution is working the way it's supposed to, right? That's good news for the elephants.
Yeah, that's right. It's great news for the elephants. It's great news for the fish, right?
Yeah.
The fish themselves, the small fish, aren't going to be targeted by the fishery, so that's awesome. So, but from our perspective, how great is it for us, and even for the future sustainability of those fish, if we're removing genetic variation, what have we done to their ability to adapt in the future?
It's also the reason why, when you look at the armor from the Greeks, like the ancient Greeks, they're tiny little men. So humans have selected them for tallness. But that's,
No, it's possible. Yeah, I don't know that study, but I wouldn't be surprised. I am way taller than anyone else in my family.
Just go to Glenbow and stand beside some of the armor.
Yeah. Oh.
You won't fit.
Cool. Yeah?
Other than the fact that, let's say that you're removing big fish and so it's not as good for humans, you don't know though what impact that the small fish is going to have in the future.
That's right.
So then they'll spin it around to you and say, well, you have no proof that that's going to be devastating, so it doesn't really matter anyway.
That's right, and this is where the precautionary principle that the DFO has in like their mandate or whatever that.
But some people don't even believe in the precautionary principle. I mean, so then you're debating a principle, you're not really debating.
You're right, and this is where science becomes so tricky too, because as Carla said, we don't prove anything in science ever. That is a philosophical underpinning of science. Is we can't prove anything. So if they ever want any proof about anything, then we're never going to get anything done, because we'll never be able to provide that sort of proof. But when we have such uncertainties in the future, you're right, it becomes a significant difficulty.
But wouldn't you say you prove it until it's unproven?
No, you get like a null hypothesis, and then you do everything in your power to disprove that, or to find evidence to reject that null hypothesis. But you never accept the alternative. You just reject the null. And even if you don't reject the null, you can't say it's not really rejected, because you might reject it in the future. This is where science becomes a little philosophically tricky, especially when we want firm proof as policy makers.